Hi, I'm Paige Wilson, a graduate student at the University of Washington in Seattle. I am a paleobotanist, so I study plant fossils, and I'm particularly looking at plant fossils from the Cretaceous and the Paleogene, which is a time period about 66 million years ago. So I work with other instructors at the Dig Field School to look at the rocks and plant fossils from Montana from this time period to understand a little bit more about what the ancient environment used to look like.
In this video, we'll talk about where the Hell Creek Research Area is, the geology of this area, and why this area is important to paleontology researchers. The Hell Creek Research Area is located in eastern Montana. This region is important to paleontologists because of the age and type of rocks that are exposed in this area. If we take a look at this geologic map, we can see that rocks exposed in eastern Montana are both Cretaceous and Paleogene in age. The Cretaceous is the period in Earth's history that lasted from 145 million years ago to 66 million years ago. This time is commonly known as the Age of Dinosaurs, although other animals like fish, other reptiles, and mammals lived during this time too. The Paleogene is the period that lasted from 66 million years ago to 23 million years ago. This period marks the beginning of the Age of Mammals, although again, other animals also lived during this period. These periods are separated by the Cretaceous-Paleogene, or KPG, mass extinction event. More specifically, here in green are the Upper Cretaceous rocks of the Hell Creek Formation. Here in yellow are the Paleocene rocks of the Tulloch member of the Fort Union Formation. There are lots of ways rocks can get to where we find them. Some are moved around on tectonic plates, some are expelled by volcanoes, some are moved by ice and water. The rocks of the Hell Creek and Tulloch formations were deposited in essentially the same places we find them today. We can see a large amount of the rocks of these formations because of erosion. The rocks of the Hell Creek Formation were deposited from about 68 to 66 million years ago, and were the last rocks of the Cretaceous period to be deposited before the KPG mass extinction. In the field, these sediments are drab and somber looking colors. Think dull green, tan, and brown. They were deposited mostly by large freshwater rivers, but also might be the remains of ponds and other wetland settings. Geologists who study these rocks can distinguish which rocks were deposited by rivers, ponds, swamps, and other environments. These observations help us to piece together what this ancient landscape must have looked like. Here is a reconstruction of what the environment probably looked like during the deposition of the Hell Creek Formation. Notice all of the dinosaurs and the mammals and insects too. The Tulloch was deposited directly after the KPG mass extinction event and was deposited 66 to 65 million years ago in the Hell Creek study area. This formation consists of thin, vibrant, and colorful rock layers and coal seams, which stand out as bright tan, white, and black bands. These sediments were also deposited by rivers, but these rivers were running through more swampy environments. Here's a reconstruction of what the environment probably looked like during the deposition of the Tulloch. No flying dinosaurs here, but lots of different mammals. Researchers at the Burke Museum and around the world are interested in studying the animals, plants, and ecosystems surrounding the KPG boundary because this boundary marks Earth's most recent mass extinction event. The KPG mass extinction event occurred 66 million years ago and killed an estimated 76% of all species, including all non-flying dinosaurs like T-Rex and Triceratops, and many other species of plants and animals. By recording the patterns of the fossil record and geologic record surrounding the KPG boundary, researchers hope to better understand how mass extinctions occur and how species survive, adapt, and evolve after them. There are not many places where rocks from before, during, and after the KPG mass extinction are found in sequence together. The Hell Creek Research Area is one of those few places, making it an important and unique place to collect paleontological and geological data. Professor and Burke Museum curator Greg Wilson Mentia and his lab visit the Hell Creek area each summer. They collect fossils from known locations that produce fossils, also called localities, prospect for new fossil localities, and collect geologic data to understand how these fossil localities are related in age to one another. The Wilson Mentia lab primarily focuses on collecting vertebrate microfossils. Next, check out the microfossils video to learn more about what microfossils are and why they are important for paleontological research.
everyone, my name is Alex Brannick. I'm a graduate student at the University of Washington here in Seattle. I'm a vertebrate paleontologist. I actually study the relatives of marsupials from the Cretaceous age. I've done a lot of microfossil work, and I've been an instructor for the DIG Field School program throughout my graduate career, and we're going to take you through the process of finding microfossils and what we do with them.
In this video, we'll talk about what microfossils are and their importance in the fossil record. Microfossils are small fossils that usually require magnification to study. They are often the leftover hard parts of something that was once alive, like teeth, skeletons, shells, and pollen. Microfossils can be as small as 1 micron, less than the width of a hair, like the tiny aquatic forams that a group of researchers here at the Burke study, but they are usually not bigger than 2.5 centimeters. That's about the size of a quarter or smaller. Any fossil that is bigger than this is called a macrofossil, like a big T-Rex skull. Small microfossils typically only represent a part of an organism and may not even be that whole part. Microfossils are usually found embedded in rock or as loose particles like pebbles or sand in deposits left over by ancient rivers. The study of microfossils and macrofossils both have challenges and benefits. Both types of fossils can give us clues about how ancient plants and animals related to each other and changed over time. Macrofossils are captivating and reveal important information about one specific species in an ancient ecosystem. We can learn a lot of different things about a particular animal's ecology from macrofossils. The ecology of an animal is how that animal interacts with other organisms and its environment, like what it eats, how it moves, and what times of the day it was awake. Macrofossils can even tell us things like if an animal experienced an injury. However, macrofossils are not as common as microfossils and usually only give us information about one species in the entire biological community or ecosystem. Microfossils can provide us data about an animal's ecology, but they can also provide us with a more complete view of an ancient ecosystem. Because microfossils are more abundant in the rock record than macrofossils, they are better for helping to see changes in ecosystems through time. Yet, because most microfossils are just tiny fragments of once living things, drawing conclusions from these fragments can be difficult. Both macrofossils and microfossils collected from the Hell Creek study area can help us learn about how ecosystems changed over time and across the KPG boundary. The fossils in the Hell Creek study area are collected on lands that are the traditional territory of the Fort Belknap, Assiniboine, and Gros Ventre tribes and Fort Peck, Assiniboine, and Sioux tribes in present-day Montana. Today, these lands are regulated by a number of different government agencies. Researchers, including those from the Burke Museum, have to get proper permits and permission to access fossil sites and collect specimens. The fossils that are collected are housed in the Burke Museum. The Burke Museum stands on the lands of the Coast Salish peoples whose ancestors resided here since time immemorial. Many indigenous peoples thrive in this place, alive and strong. The Burke Museum is designated as a federal repository, which means that any fossils collected on federal land, even outside of Washington state, can be kept in the collections at the Burke. The Burke Museum plays an important role in preserving these specimens and making the data available to researchers and the public forever. Preserving fossils and the data we gain from them is critical for ongoing and future scientific research. Today, the Burke holds over 20,000 vertebrate fossils from the Hell Creek study area. Let's talk about the microfossils you will be seeing and how to find them in the next video.
Hi, here we are going to go over the process of sorting microfossils from a bag of sediment. The tools you will be using for this are a bag of fossiliferous sediment, a tablespoon scoop, fine paintbrushes, sorting trays, magnifying glasses, some sort of collection material for your found fossils, and your ID guide or identification guide. First, let's talk about the bag of fossiliferous sediment that you will be using. One way in which microfossils are collected is through bulk sediment collection. Researchers in the field will find the layer or horizon where microfossils are being found and will collect bags of loose sediment from that fossiliferous horizon. These bags of fossiliferous sediment are then screen washed to separate out the rocks and other bits of sand particles and non-fossil material from the fossiliferous sediment, which gets concentrated and put in Ziploc bags. After screen washing, we end up with smaller bags of sediment like this. Each bag is systematically labeled with identifying information telling us where that bag came from. Each bag is labeled with two parts, a locality name and a field number. Each field number has several parts to it. First are the initials that refer to the person who found it. Second is the HC alpha code, that means Hell Creek Project. Third is a set of numbers referring to the year, 2018 in this case. And fourth is a number referring to the collection event in that year. Each member of the collecting team will also use this field number in their field notebooks and in all of their records to keep track of exactly when and where this sediment was collected. The fossils that are eventually found in this bag of sediment will each get a unique identifier or specimen number at the Burke Museum, but the field number and the field name, locality name, will be kept in all records and notes referring to the specimens and fossils that are found inside of this bag.
So now we can move on to the actual sorting process. The first thing I'm going to do is I'm going to take a tablespoon, scoop some sediment out of our bag, and then carefully spread it across our tray. So it's really important that we have the right amount of sediment in our sorting tray. Too much sediment can actually make the sorting process difficult, time-consuming, and then we'll probably miss fossils. So now that I have this tablespoon scooped out into the tray, I actually want to use the paintbrush to carefully spread it out. So once I've spread this sediment out, it will make it much easier to look through the sediment for these microfossils. So if we take a look here, this is about the right amount of sediment we want in our tray. So to start looking for fossils, what I'm going to do is I'm going to take my magnifying lens or hand lens. With these, you actually want to put the hand lens to your face first and put it up to your eye. We're going to start sorting from the bottom left corner. And we're going to go through a zigzag pattern across this tray and pick out every microfossil that we find. So we'll go ahead and start here.
So you have to get very close, and we'll just carefully and slowly go through the sediment. And I'm just going across to the right. I don't see anything quite yet.
Okay, so I think I found a microfossil. So what I'm going to do is I'm going to take our paintbrush, carefully spread it out to the side. And these microfossils are really delicate, so we want to be very careful with them. And I'll try and scoop it up with our paintbrush. If you can't do that, that's okay. We just want to make sure that that fossil's out of the way, and we can use our hands, but again, very carefully, to pick it up and put it in our collection tray. So I've picked out one fossil here. So now what I can do is I can continue from where I left off and continue searching for fossils.
So I'll just go back across the tray. Again, you want to do this very slowly and very carefully.
And we want to continue this process until we get through the entire tray. Recognizing fossils takes a lot of practice, so don't get discouraged if you don't find anything right away. Fossils have a number of characteristics that you can look for to help pick them out of your sediment. For example, bone is really porous, so it'll have a lot of little holes in it. Teeth and scales are dark and shiny in color, and they're usually darker than the sediment. And fossils, most of all, have recognizable shapes. So they have patterns that you can look for in which the sediment around it won't have. So you can go ahead and take a look at the example microfossils to help you decide and figure out what you're looking for in the sediment. So a lot of these microfossils are really tiny, so it's important to go through this very slowly and carefully and keep your eyes out. And most of all, definitely always pick out something that, even if you're not sure if it's a fossil or not, it's better to pick it out than to accidentally leave an important fossil behind. Okay, so once you've searched through your tray of sediment a few times and you've picked out everything that you can find, you can go ahead and put your sediment in the discard pile and grab another tablespoon of sediment to sort through. After you're finished sorting, we can then move on to actually identifying the fossils that you found.
Hi, in this video we're going to talk about the process of identifying or IDing the fossils that you have found from your sediment.
Before you start identifying your microfossils, get familiar with your sorting tray. Place your unidentified fossils here. This is also where you can leave fossils you can't identify with the dichotomous key. If you're using the Advanced ID Guide, you'll use your sorting tray this way. You'll sort your fossils into Mammalia, Chondrichthyes, Amphibia, Osteichthyes, Reptilia, and fish scale categories. If you're using the Beginner ID Guide, you'll use your sorting tray this way. You'll sort your fossils into the green fish scales, blue bones, and red teeth categories. The dichotomous key will tell you which category your fossil belongs in.
Now take a look at the fossils that you found. What do you notice about them? How could you describe some of them? What do you notice about their shape and texture? And what can you see that makes certain fossils look similar to each other and others look different? What makes them unique? So let's look at each fossil one at a time. And we'll be using our dichotomous key to help guide us and determine what type of fossil it is.
Okay, so let's go ahead and look at this example here of a microfossil that I have. What are some ways that we can describe this fossil? Okay, so I'm going to start by making observations of this microfossil. The first thing I can see about it is that it's dark in color, and that it also has this shiny texture to this side of it. I can also notice that it's pretty flat. There's not a lot of thickness to this fossil. And on the other side, it's a different color. It's not as shiny. Lastly, I can see that the overall shape of this fossil is what I would consider to be diamond-shaped. Okay, so using the observations that I made about the fossil, we're then going to identify this fossil using the Microfossil ID Guide. So we'll start with the first question. And the first question within this fossil ID guide is the fossil mostly flat? So looking back at our microfossil, I would say yes, the fossil is mostly flat. So we'll go ahead and click this for the next question. The next question is, what best describes your fossil? Is it thin with smooth edges, or is it not thin and it has edges that has a spongy texture to it? If I look back at my fossil, I can see that it has smooth edges and that it's thin. So we'll go ahead and click that one. So our next question that our ID guide has brought us to says, is the fossil smooth and shiny like glass on one side and dull on the other? If we go back and look at our fossil, yes, that's true. On one side it's shiny and like glass, and on the other side it's dull. So we'll go ahead and click yes. Our next question is the fossil shaped like a diamond? And yes, it is. So if we look back at our fossil, yes, it's shaped like a diamond. And then it brought it to our answer. So it looks like we have a Gar scale. It's important to keep in mind that a lot of the microfossils that you might find may only be a part of the entire piece. So over here is the Gar scale that we looked at before, and it's a complete Gar scale. And over here is just a fragment of a Gar scale. But although this one is not completely diamond-shaped, they still share a lot of characteristics. So for example, this fragment of agar scale is still shiny on one side and dull on the other, just like our complete gar scale. Additionally, it's still pretty flat and thin with smooth edges. It's not very thick. So although it's not a complete diamond shape, we can still classify this as a gar scale.
Now we know this fossil is a gar scale. If you're using the Advanced ID Guide, the gar scale will go in this fish scales category right here.
So once I've identified all of the fossils that we found in that sediment, we'll go ahead and put those in those collection containers that you have. Make sure that they're labeled and tightly closed. Those collection containers will then come back to the Burke Museum and be incorporated into the collections to be used by researchers. Thanks for joining us on our microfossils video series. Good luck searching for those fossils, and we're excited to see what you find.